Enzymes were discovered by Edward Buchner, but the term enzyme was given by Frederick W. Kuhne. The isolation and crystallization of urease by James Summer in 1926 was a breakthrough in early enzyme studies, and he postulated that all enzymes are proteins. However, ribozyme and ribonuclease P are non-protein enzymes. Enzymes are biological catalysts that increase the speed of a chemical reaction. Although enzymes influence a chemical reaction, it is not itself permanently changed, nor does it cause the reaction to occur. Thus, a catalyst can increase the speed of a reaction, but cannot cause the reaction and can be reused again as they are not used up in the reaction. Enzymes are organic catalysts produced by living organisms. The reactant in an enzyme catalyzed reaction is called the substrate. The active site of an enzyme is that small portion of the molecule which is responsible for the catalytic action of the enzyme. Enzymes provide a chemical pathway that has a lower activation energy that uncatalyzed reactions do not have. Enzymes are usually much larger than their substrates. The sizes ranges from just 62 amino acid residues as in 4-oxalocrotonate tautomerase to over 2500 residues in fatty acid synthase. Only a small portion of their structure is directly involved in catalysis and is called the catalytic site. This catalytic site is located next to one or more binding sites where residues orient the substrates. The catalytic site and binding site together comprise the enzyme's active site. The remaining majority of the enzyme structure serves to maintain the precise orientation and dynamics of the active site. Enzymes are generally globular proteins acting alone or in larger complexes. Like all proteins, enzymes are linear chains of amino acids that fold to produce a three-dimensional structure. The sequence of the amino acids specifies the structure, which in turn determines the catalytic activity of the enzyme. Although structure determines function, a novel enzyme's activity cannot yet be predicted from its structure alone. Substrate binding. Enzymes must bind the substrates before they can catalyze any chemical reaction. Enzymes are usually very specific as to what substrates they bind, and then the chemical reaction is catalyzed. Specificity is achieved by binding pockets with complementary shapes, charge and hydrophilic or hydrophobic characteristics to the substrates. Enzymes can therefore distinguish between very similar substrate molecules to be chemoselective, regioselective and stereospecific. Enzymes have varying degrees of specificity. Some enzymes have absolute specificity for one substrate and none others, while other enzymes react with substrates with similar functional groups, side chains or positions on a chain. The least specific enzymes catalyze a reaction at a particular chemical bond regardless of their structural features. Some of the enzymes showing the highest specificity and accuracy are involved in copying and expression of the genome. Some of these enzymes have proofreading mechanisms. Here, an enzyme such as DNA polymerase catalyzes a reaction in a first step and then checks the product that is correct in a second step. This two-step process results in an average error rates of less than 1 error in 100 million reactions in high fidelity mammalian polymerases. Similar proofreading mechanisms are also found in RNA polymerase, amino acyl tRNA synthetase and ribosomes. Conversely, some enzymes display enzyme promiscuity having broad specificity and acting on a range of different physiologically relevant substrates. Enzyme changes shape by induced fit upon substrate binding to form enzyme substrate complex. Hexokinase has a large induced fit motion that closes over the substrate adenosine triphosphate and xylose. In 1894, Emil Fischer proposed that both the enzymes and substrates possesses specific complementarity geometric shapes that fit exactly into one another. This is often referred to as the lock and key model. This model explains enzyme specificity but fails to explain the stabilization of transition state that enzymes achieve. The induced fit model. In 1958, Daniel Koshland suggested a modification to the lock and key model. Since enzymes are rather flexible structures, the active site is continuously reshaped by interactions with the substrate as the substrate interacts with the enzyme. As a result, the substrate does not simply bind to a rigid active site. The amino acid side chains that make up the active site are molded into the precise positions that enable the enzyme to perform its catalytic function. In some cases, such as glycosidases, the substrate molecule also changes shape slightly as it enters the active site. The active site continues to change until the substrate is completely bound, at which point the final shape and charge distribution is determined. Induced fit may enhance the fidelity of a molecular recognition in the presence of competition and noise via conformational proofreading mechanisms. The basic mechanism by which enzymes catalyze chemical reactions begin with their binding of the substrate to the active site on the enzymes. The active site is the specific region of the enzyme which combines with the substrate. The binding of the substrate to the enzyme causes changes in the distribution of electrons in the chemical bonds of the substrate and ultimately causes the reaction that lead to the formation of products. The products are released from the enzyme surface to regenerate the enzyme for another reaction cycle. An enzyme combines with substrate to form a short-lived enzyme-substrate complex. Once a reaction has occurred, the complex breaks up into products and enzyme. Thus, the enzyme remains unchanged at the end of the reaction and is free to interact again with more substrates. The active site has a unique geometric shape that is complementary to the geometric shape of a substrate molecule, similar to the fit of puzzle pieces. This means that enzymes specifically react with only one or a very few similar compounds. The enzyme lowers the activation energy of reaction, that is, the energy required for substrates to react in order to get converted into product. It is believed that enzyme activity occurs in two steps. First, the active site of the enzyme combines with the substrate to form an enzyme substrate complex. Second, this enzyme substrate complex then breaks up to form the products and the free enzyme, which can react again. According to the lock and key model, the substrate must fit into the active site of the enzyme, hence the specificity of that enzyme. For substrates that have an optically active site, it is believed that there must be 3 points of attachment between the substrate and the active site of the enzyme, so that only one of the two optical isomers fits. In the body, enzymes are specific for L amino acids and D carbohydrates. According to induced fit model, the active site is not rigid as in the lock and key model, but flexible. That is, the site changes its conformation upon binding to a substrate in order to yield an enzyme substrate fit. Enzymes can accelerate reactions in several ways, all of which lower the activation energy. By stabilizing the transition state, it creates an environment with a charge distribution complementary to that of the transition state of its lower energy. By providing an alternative reaction pathway, it temporarily reacts with the substrate, forming a covalent intermediate to provide a lower energy transition state. By destabilizing the substrate ground state, by distorting bound substrates into their transition state form to reduce the energy required to reach the transition state. By orienting the substrates into a productive arrangement to reduce the reaction entropy change. However, the contribution of this mechanism to catalysis is relatively small. Enzymes may use several of these mechanisms simultaneously. For example, proteases like trypsin perform covalent catalysis using a catalytic triad, then stabilize charge built up on the transition states using an oxyanion hole, and finally complete hydrolysis using an oriented water substrate. Acid-base catalysis occurs by proton transfer. General acid catalysis is a process in which proton transfer from an acid lowers the free energy of a reaction's transition state. Bovine pancreatic RNase provides an example of enzymatically mediated acid-base catalysis. Covalent catalysis usually requires a nucleophile. Covalent catalysis accelerates reaction rates through the transient formation of a catalyst substrate covalent bond. Usually, this covalent bond is formed by the reaction of a nucleophilic group on the catalyst with an electrophilic group on the substrate, and hence this form of catalysis is often called nucleophilic catalysis. Covalent catalysis can be conceptually decomposed into three stages. The nucleophilic reaction between the catalyst and the substrate to form a covalent bond. The withdrawal of electrons from the reaction center by the now electrophilic catalyst. The elimination of the catalyst, a reaction that is essentially the reverse of stage. Metal ion cofactors act as catalyst. Nearly 1/3 of all known enzymes require metal ions for catalytic activity. This group of enzymes includes the metalloenzymes, which contain tightly bound metal ion cofactors, most commonly transition metal ions such as ferrous ion, ferric ion, cuprous ion, manganese ion or cobalt ion. These catalytically essential metal ions are distinct from ions such as sodium ion, potassium ion or calcium ion, which often play a structural rather than a catalytic role in enzymes. Ions such as magnesium ion and zinc ion may be either structural or catalytic in nature. Metal ions participate in the catalytic process in three major ways: by binding to substrates to orient them properly for reaction, by mediating oxidation-reduction reactions through reversibly changes in metal ion oxidation state, by electrostatically stabilizing or shielding negative charges. Catalysis can occur through proximity and orientation effects. The efficiency of enzymes arises from the specific physical conditions at enzyme catalytic sites that promote the corresponding chemical reactions. The most obvious effects are proximity and orientation. Reactants must come together with the proper spatial relationship for a reaction to occur. By simply binding their substrates, enzymes facilitate their catalyzed reactions in 4 ways. Enzymes bring substrates into contact with their catalytic groups and in reactions with more than one substrate with each other. Enzymes bind their substrates in the proper orientation for reaction because molecules are not equally reactive in all directions. Rather, they react most readily if they have the proper relative orientation. Charged groups may help stabilize the transition state of the reaction, a phenomenon termed as electrostatic catalysis. The charge distribution around the active site of enzymes may also guide polar substrates towards their binding site. Enzymes freeze out the relative translational and rotational motions of their substrates and catalytic groups. This is an important aspect of catalysis because in the transition state, the reacting groups have little relative motion. Enzymes catalyze reactions by preferentially binding the transition state. An enzyme may bind the transition state of the reaction it catalyzes with greater affinity than its substrates or products. When taken together with the previously described catalytic mechanisms, preferential transition state binding explains the observed rates of enzyme catalyzed reactions. The original concept of transition state binding proposed that enzymes mechanically strained their substrates towards the transition state geometry through binding state into which undistorted substrates do not fit properly. The original concept of transition state binding proposed that enzymes mechanically strained their substrates toward the transition state geometry through binding site into which undistorted substrates did not fit properly. Such strain promotes many organic reactions. The more tightly an enzyme binds its reaction's transition state relative to the substrate, the greater is the rate of the catalyzed reaction relative to that of the uncatalyzed reaction. That is, catalysis results from the preferential binding and therefore the stabilization of the transition state relative to the substrate. Enzymes are not rigid, static structures. Instead, they have complex internal dynamic motions. That is, there are movements of parts of the enzyme's structure such as individual amino acid residues or groups of residues forming a protein loop or unit of secondary structure or even an entire protein domain. These motions give rise to a conformational ensemble of slightly different structures that interconvert with one another at equilibrium. Different states within this ensemble may be associated with different aspects of an enzyme's function. For example, different conformations of the enzyme dihydrofolate reductase are associated with the substrate binding catalysis, cofactor release and product release steps of the catalytic cycle. Allosteric modulation. Allosteric sites are pockets on the enzyme distinct from the active site that bind to molecules in the cellular environment. These molecules then cause a change in the conformation or dynamics of the enzyme that is transduced to the active site and thus affects the rate of reaction of the enzyme. In this way, allosteric interactions can either inhibit or activate enzymes. The allosteric interactions with metabolites either upstream or downstream in an enzyme's metabolic pathway are influenced by feedback regulation. This alters the activity of the enzyme according to the flux through the rest of the pathway. Temperature. The speed of all chemical reactions is affected by temperature. The higher the temperature, the faster is the rate of reaction. This is also true for reactions involving enzymes. However, if the temperature is raised too much, the enzymes will be inactivated by denaturation and will be unable to function. The best temperature for enzyme function is the temperature at which the rate of a reaction involving an enzyme is the greatest and is called the optimum temperature for that particular enzyme. Role of pH. Each enzyme has a pH range within which it can best function. This is called the optimum pH range for that particular enzyme. For example, the optimum pH of pepsin, an enzyme found in gastric juice, is approximately 2, whereas the optimum pH of trypsin, an enzyme found in pancreatic juice, is nearly 8.2. If the pH of a substrate is too far from the optimum pH required by the enzyme, that enzyme cannot function at all. However, since body fluids contain buffers, the pH usually does not vary too far from the optimum values. Effects of concentrations. As with all chemical reactions, the speed is increased with an increase in concentration of reactants. With an increased concentration of substrate, the rate of the reaction will increase until the available enzyme becomes saturated with substrate. Also, with an increase in the amount of enzyme, the rate of reaction will increase, assuming an unlimited supply of substrate. Inhibitor. An enzyme inhibitor is any substance that will make an enzyme less active or render it inactive. Enzyme inhibitors that bind reversibly to the active site and so block SS by the substrate are called competitive inhibitors. Their effect can be overcome by increasing the concentration of the substrate. Other inhibitor that binds to another site of the enzyme to render it less active or inactive are called non-competitive inhibitors. They act by changing the conformation of the enzyme, thereby reducing or stopping its activity. Irreversible inhibitors form strong covalent bonds with the enzyme, rendering it inactive. This effect cannot be overcome by increasing the concentration of the substrate. Enzymes are proteins that catalyze biological reaction and make them faster by reducing the activation energy by induced fit mechanism. Substrate molecules bind themselves at the active site of enzyme to form the products. We can measure enzyme activity by either measuring appearance of products or disappearance of substrates. Substrate initially binds to the enzymes by non-covalent interaction, including hydrogen bonds, ionic bonds, and hydrophobic interactions. Enzyme activity is affected by concentration of enzyme, substrate, temperature, pH, water activity, and inhibitors. Enzymes have many applications in industry and in food processing as well as preservation.